The UK COVID variant has been successfully cultured and isolated in the National Institute of Virology in Pune. You're watching TV9 News and I'm Prajwal. The Indian Council of Medical Research announced that the new coronavirus variant that was found in the United Kingdom has been cultured and isolated in the National Institute of Virology in Pune through samples collected from returnees from the United Kingdom. The scientists also used vero cell lines for the successful culture of the virus. So why is this important? To tell you this, I'm joined by Dr Sitesh Roy. Dr. Roy, what is the significance of developing the new variant in a lab?
So Prajwal, once we have cultured a virus in the lab, we can do a lot more in terms of research as well as vaccine development. In addition to studying and understanding the virus structure, differences, etc. through that kind of a culture, and hence having a live culture of the new variant is an advantage for us to do all of these things.
What is cell culture and why is it important?
So cell culture is the process in which we are able to grow the virus in vitro, which means in a petri dish with the right kind of temperature, nutrition, circumstances in which the virus thrives, grows and propagates, so that that can then be used for studying its structure, studying its behavior and function, studying its transmissibility, and also using it to make new vaccines if that is ever necessary.
How will this prove to be important in understanding the efficacy of vaccines against new strains and variants?
So whenever we have a new strain or a variant, structurally knowing the differences between that strain or variant and the existing vaccine, the spike proteins of that vaccine are really the main process of knowing what the efficacy of that old vaccine will be with the new variant. And so having the structure, the genetic makeup, having the shape and structure of the spike proteins, all of these will help researchers and scientists better delineate whether the new variant will still be susceptible to the old or the previous vaccine that has been developed.
What are vero cell lines that are used in the successful culture of the virus?
A vero cell line is a cell culture that was derived from the kidney of an African green monkey. And this cell line has been internationally used for decades now to culture and grow viruses in a petri dish or in vitro. And this usually requires a lot of specific substances, including fetal bovine serum, which is a very important ingredient in keeping these vero cell lines alive. And on these cell lines, the virus is grown, cultured and studied.
What forms of research can be done on the viruses cultured in a lab?
So right from finding out if there is a new antiviral drug which can work in either suppressing or killing a virus in the petri dish in the lab, to actually identifying ways in which the virus spreads or attacks different types of cells, to identifying the specific vaccine targets that would best work against the virus. There is a ton of information that can be generated from culturing viruses in a lab. And so all of this becomes practical and possible when we have that cell culture with a live virus in it to study.
The Indian Council of Medical Research has claimed that no country has yet reported the successful isolation and culture of the UK variant. The virus culture will also be useful in conducting research on the possibility on other forms of outbreaks. If you like this video, do click on the like button and don't forget to subscribe to TV9 News on all the social media platforms. Thank you for watching and have a great day.